I don't know if I can. Okay. All right, so first, what I want to do is, I'm having trouble finding where I can actually share my screen.
There's a share screen.
Right, I see it. I see it. I got it. I'm sorry. I share this one. And before we get started, what I want to do is give a quick shout out to the Delaware Valley Mass Spec discussion group, which is organizing this, obviously, and you're all here because you were invited. But before we start, what we want to say is that we're ramping up our sponsorships from everyone that was helping us out prior to COVID, and we'd like to return to normalcy as fast as possible, and this requires some funding and getting back to speed where we were before everything. So if you were a vendor before that was sponsoring us with dues normally for the year, we ask that we get back on that, and we'll be circulating some letters that start to show that. And specifically who we're talking is Mobileye, Agilent, Bruker, Gentek, IonSense, Geol, Keystone, LECO, New Objective, NEMS, Phoenix, S&T, Restex, SciEx, Shimadzu, Adaptis, Thermo, Trajan, and Waters. And thank you to Bruker, especially, who has already come through on that and committed to funding for the next year. So with that, I'm going to.
And also, it would be nice to say that we also accept new members, right? New vendors. Anybody who wants to sponsor us are welcome to, join the, officially join the Delaware Valley sponsor list. One more thing, Joe, I think we need to show this is that we have a social media presence. So if you look at the top right-hand corner of the website, there is Indeed, LinkedIn. What's the second one?
Instagram and YouTube.
Instagram and YouTube. Not much of an Instagram user, but definitely, YouTube has all the videos of the speakers who are willing to share their talk. We post them there. So if you missed this, or if you want to relive all the moments, crazy moments, you can go back and view that. So yeah,
right, that's where all the fun is.
Yeah.
So Boone, you can go ahead and get ready, and I'll give the introduction here. So it's my pleasure to have Boone Prentice with us. He got his BS in chemistry with honors and distinction from Longwood University in Virginia. And that was in 2008. He minored in biology and mathematics, really a jack of all trades. He did a lot of electroanalytical research using biosensors under Melissa Roten, and then attended graduate school with Scott McLuckie, obviously at Purdue. With Scott McLuckie, he did tons of really cool research that we're just talking about that comes from ion-ion reactions, real analytical instrumentation focused work. And after his PhD with Scott, he moved on to Richard Caprioli's lab at Vanderbilt in Nashville. And there is where he started doing, looks like a lot of matrix-assisted laser desorption, ionization, imaging. And this is going to be kind of the focus of the talk today, and specifically FTICR and TOF systems, and imaging mass spec to study diabetes, cancer, drug delivery, and infectious diseases. So I'm looking very forward to the talk today, high chemical resolution imaging mass spec using gas phase ion-ion reaction. So please take it away.
Thank you, Joe, for that introduction and the opportunity to speak here with you guys today. I'm excited to tell you about what we've done a little bit in my group over the past couple of years since arriving at UF. So I like to start off by putting our work in context of other biological imaging modalities. And if we consider the general field of biological imaging, despite what we might think with all the recent awesome technological advances, this certainly isn't a new field. So it probably dates back to maybe the late 1600s or so when Van Leeuwenhoek first developed the light microscope. And over the past 350 years or so, you can see that the technology has certainly advanced tremendously. So on the right is a picture of Eric Hetzig's Palm device, for which he won the Nobel Prize in chemistry in 2014 for super resolution microscopy. And this is a really cool story. If you're not familiar with Hetzig's work or his story, he basically built this device while he was technically unemployed in his living room. So a really fascinating story of scientific hard work, preservation, and ingenuity. But perhaps, I'm an instrumentalist. I think things like this are pretty fascinating, but perhaps more important than the advances in the instrumentation and technology is the advances in the types of images we're able to acquire with this technology. So on the left is a notebook sketch of some of the microbes that Van Leeuwenhoek was observing under his light microscope. And on the right is a picture of neuronal activity in a zebrafish brain acquired with the Palm device. And so you can see that obviously the amount of informational content that we're able to acquire or glean from these images has advanced tremendously. And that's really important because one of the easiest ways we have for understanding complex systems and one of the easiest ways we have for conveying that understanding to other individuals and scientists is through the use of images. And so if we consider the general suite of imaging modalities, I've listed a few of them here, and these run the gamut of things that are pretty common in everyday parlance, and at least in terms of maybe a doctor's office, things that we might encounter like an ultrasound or an X-ray or even an MRI, to things that maybe kind of are still in the clinical setting, but maybe a little bit more in the research lab space, or at least the lab space of a clinical setting, things like maybe stain microscopy or PET, things that require a little bit more training or experimental workflows that are a little bit more detailed to consider, to things that are definitely probably fall into, at least currently, much more of the research space and are just starting to make their way across that translational bridge, things like, certainly imaging mass spectrometry. And what I think in particular that imaging brings is a really high degree of molecular specificity. So each of these has their merits and their challenges associated with them. So some of these are capable of making in vivo measurements, so measurements while the subject is still alive. Others require antibody tags or radio tags that could be a detriment to the experimental workflow. And still others are capable of very high spatial resolution, so cellular or subcellular spatial resolution. But again, the strength that imaging mass spectrometry or mass spectrometry imaging brings is this really high degree of molecular specificity. So we're leveraging the molecular specificity of the mass spectrometer with the microscopic imaging approach. And there's obviously a few different flavors of imaging MS that you could consider, things like MALDI, DESI, and SIMS, and I'll be largely talking about MALDI here today. So this general workflow, I won't spend a ton of time on this. You all might be familiar with the general procedure of how we do these images, so I'll just kind of go over this very briefly. Typically, at least in my lab, we're looking at biological tissue specimens. So we prepare thin sections of those specimens on something like a cryomicrotome and mount them to a flat substrate. We then apply a MALDI matrix, which is just going to be a small organic acid that's designed to absorb the wavelength of our incident MALDI laser. Obviously, MALDI stands for matrix assisted laser desorption ionization. And we perform a raster of the tissue surface with our MALDI laser, and we ablate material at each position and sample that into the mass spectrometer. So each position allows us to generate a spectrum for the informational or molecular content in the tissue at that position. And that after we've acquired tens of thousands of spectra across the entire tissue section, we can reconstruct heat maps of ion intensity for any individual ion of interest. So we can do this for proteins, peptides, lipids, pharmaceutical compounds, et cetera. And so here's a couple kind of images that I think faithfully represent the general state of the art of imaging mass spectrometry in the field. On the left, you can see a single hemisphere or one half of a transverse rat brain section. And each color in this image is a different individual lipid or protein or metabolite. In this case, they're lipids. And the relative brightness of that color corresponds to the intensity or the abundance of that compound in the tissue. And so I think that this is a really nice high spatial resolution image. You can even kind of make out in yellow here in the cerebellum, the Purkinje cell layer, which is a single cell layer in the brain. And when we show this image to some of our collaborators, they think on first pass or first inspection that this is a fluorescent image, because I think the level of kind of dynamic range and structural detail in the image just looks really good. But obviously, we don't have the spatial resolution or sensitivity of fluorescence imaging, but the amount of pixels that we're able to acquire in this image can make that image really kind of pop. In terms of the spatial resolution that we can actually achieve, on some home-built systems, during my postdoc, we had an instrument that was capable of around a micron spatial resolution, and so this is an example acquired on that platform. So this is a human kidney glomerulus, shown in red, surrounded by proximal and distal tubules in aqua and blue. And if you look in some of these tubules, you'll see these little black flecks, and those actually aren't tissue aberrations. When you compare that to an H&E stain, those are actually individual cell nuclei. And so you're starting to resolve some subcellular structures. This is kind of what I would call a more heroic experiment, at least when it was acquired maybe 5 or 10 years ago. But it demonstrates that we are capable of making these types of measurements in the field. Additionally, now that we have lasers and systems that are capable of much higher throughput than they were 20 years ago, we're able to acquire much larger regions of tissue and at higher spatial resolution. So some of the lasers nowadays are operating at 10 and 20 kilohertz, and especially on time of flight mass analyzers, we're able to acquire data relatively rapidly. So we can sample over 1 million pixels, for example, as shown on this human lung, or a full body cross section of a mouse pup. So the throughput of the instruments nowadays is allowing and enabling different types of really impressive experiments to be performed. And then obviously we can apply and use imaging mass spectrometry to study a whole bunch of diseases, biological systems, tackle a whole bunch of clinical challenges. And so in my lab, we're doing things like studying infectious disease, studying diabetes, studying cancer, and studying drug distributions and quantification. So I won't talk a ton about those applications here, but just know, and I'll try and highlight near the end, that we're hoping to develop tools that allow us to study these disease systems better and make better inferences into the underlying tissue biochemistry that we're trying to study. So one key part of this experimental workflow that I left out is identification. And so obviously, if you're doing mass spectrometry, this is not new to you, but I think doing this in an imaging mass spectrometry workflow introduces some unique challenges. So the way that we typically do identification in imaging currently is we use maybe some exact mass measurements using a high resolution mass analyzer. We can also do some off tissue, kind of offline LC-MS/MS experiments. So basically we take the tissue section, homogenize it up, and analyze it via classical LC-MS/MS. But obviously we've lost the spatial component to that measurement, and so it can be difficult to link our chromatographic experiment with our imaging experiment. And then we can perform some on-tissue tandem mass spectrometry. And so I think that's a really nice experiment, but it's not always successful, or it doesn't always give us the information we like, at least if we're using classical collision-induced dissociation. So again, this is a unique challenge in imaging, and the real reason for this is because of the complexity of the tissue environment. So what I'm showing here is the average mass spectrum from a rat brain tissue section, that rat brain that I showed a couple slides earlier. And this was acquired on an FTICR platform. And so you can see that there's maybe a few dozen peaks here, but we know this is an ICR. So obviously if we blow up the y-axis, you can see that there really are hundreds or thousands of individual peaks present. And if we zoom again in on just a single half Dalton mass range and zoom further in on the y-axis, you can see that there's maybe a dozen peaks with relatively decent signal to noise ratio. And each one of these peaks could generate an ion image of interest. So this is a mosaic of a few hundred of those individual ions. And again, each one of these could have a unique distribution, a unique biochemical functionality that's playing in the tissue. And so it's important for us to resolve and identify these. And as I mentioned, high resolution accurate mass on something like an FTICR or an Orbitrap instrument can allow us some ability to separate some of these isobaric compounds. So here's an example where we're studying a human pancreas tissue section, and we're looking at these 3 lipid ions, and they're separated. I think this is roughly a 50 millidalton mass range that I've highlighted here in red. And you can see that the phosphatidic acid, the PA38.3, is localized to the islets in this pancreas tissue specimen. And the other two lipids are localized to other regions in the exocrine tissue. And so if we didn't have the ability to resolve these 3 ions from one another, the consequence would be twofold. Essentially, we would look at a composite image of all 3 species, and so that would give us an inaccurate picture of the distribution. But then secondly, we also wouldn't be able to identify unambiguously the ion of interest. So we have a spatial component that would result in an error, and a molecular identification error that could result from that failure to resolve these components as well. And so high mass resolution can impart solve this issue. So, some of our high resolution analyzers are now capable of, in excess of 1 million resolving power that can allow us to separate some of these isobaric compounds, but this does come at a significant cost. Obviously, in terms of financial costs, our FTICR and Orbitrap platforms tend to be our more expensive platforms. Our lower cost mass analyzers, something like a quadrupole mass analyzer that you would get a resolving power around 5 to 10,000, you can see in this black trace, would be more inexpensive, but obviously not capable of the resolving power we need to differentiate these. But additionally, these high resolution platforms also come at a significant cost in terms of the time required to do the experiment. So these are FT-based analyzers. So if we want a higher resolving power, we need to record transients for longer periods of time. And so this can be 1, 2, 4, 10 seconds. And if we're doing a 10-second acquisition at every single pixel and we're requiring 10, 20, 30,000 pixels, the imaging experiment quickly becomes untenable. Additionally, all the resolving power in the world is not going to allow us to separate isomeric compounds. We need more, we need additional ways that we can enable this differentiation. Again, the consequence here is that the spatial distribution of a single peak really may not faithfully represent just a single chemical species. So in addition to accurate mass, gas phase separation using ion mobility has emerged as a really powerful tool in imaging mass spectrometry. This is great because it's rapid, but unfortunately it doesn't pretend to provide primary structure information. So we're not going to get any information about what bonds or what chemical bonds or functionalities are bonded to each other. And so for that, obviously in the field, we typically use tandem mass spectrometry, and this enables some structural information. So through piecing back together the fragment ions that we detect, we can identify the original structure of our compound of interest. Additionally, as long as the isomers fragment differently, we can differentiate or distinguish between multiple isomeric components. And then we also get a sensitivity improvement because we're eliminating chemical noise. And so here's just a quick example of a tandem mass spectrometry imaging experiment. So this is an example where Kristen Burnham-Johnson in my postdoc lab in the Caprioli lab was looking at phosphatidylethanolamin lipids in mouse embryos. And if you're not familiar with lipid nomenclature, the PE is the lipid head group or lipid class. This is the phosphatidylethanolamin. 36 is the total number of carbons in the fatty acyl tails. So the SN1 and SN2 fatty acyl tails have a total of 36 carbons, and 2 is the total number of double bonds in those carbons. And so when Kristen performed conventional tandem mass spectrometry using collision-induced dissociation, she was able to differentiate the 18-0, 18-2 from the 18-1, 18-1 isomers. And she was able to do that because they cleaved at this fatty acid position, and she was able to report obviously different fragment masses. And this had important implications in the mouse embryonic development. The 18-1, 18-1 phosphatidylethanolamin isomer was localized to the M pole, whereas the other isomer was localized kind of more ubiquitously across the tissue. And so this is an instance where CID was successful, but unfortunately, collision-induced dissociation isn't always successful in a tandem mass spectrometry experiment. So again, I think, especially in imaging mass spectrometry, and especially with phospholipids, this is a really unique challenge. In terms of imaging mass spectrometry, we obviously don't have the upfront benefit of separation, chromatographic separation like GC or LC. Again, if we're trying to perform GC or LC at every single pixel and we're doing that 10,000, 20,000 times across the tissue section, that's not practical on an imaging timescale where we want to acquire the images over 2, 3, 4, maybe 8, 12 hours or so. Additionally, we're limited to the ion types that we generate from the tissue surface. So it can be difficult to derivatize the tissue prior to analysis. And we know that there's a ton of condensed phase derivatization protocols that have enjoyed wide success in the mass spectrometry community. And there are some that are done in imaging mass spectrometry as well, but as I'll talk about, this is quite a bit more difficult than we would like. So if we consider all of the different head groups, phosphatidic acids, ethanolamines, choline, serines, inositols, glycerols, all the different head groups, all of the different fatty acyl tail lengths and orientations, are they in the SN1 or SN2 position? we consider all the number of double bonds and the double bond position and stereochemistry along the fatty acyl backbones, there's nearly 100,000 possible lipid structures that are present in our lipidome. And so accurate identification and resolution of these different chemical species is really important to understanding the biological implications. So the biochemical synthesis, the metabolism, and so on is really critical in a lot of instances. So just to kind of visualize what this challenge is. So, for each spectral peak, in general, we're going to have multiple isomers present in our biological tissue specimen. So this is an example where we're looking at PC34-1, and in this cartoon schematic, we're detecting this as a single peak. But really, we know this is a mixture of at least 2 different isomers. So I'm showing 2 isomers here, one where we have the double bond in the SN1 position and one where we have it in the SN2 position. And this precursor MS1 peak, this PC34-1 peak, is really a combination of multiple isomers. And so we need ways of resolving this. And so our approach is to use derivatization. And as I mentioned before, derivatization has a long history in mass spectrometry. It's been used for decades. And it's also seen interest in the imaging mass spectrometry community over the past decade or 2. And conventionally, that experiment proceeds by applying the derivatization agent ubiquitously across the tissue surface using something like a robotic sprayer. And then we incubate the tissue for a certain period of time. This could be minutes or even hours. And then we perform our MALDI analysis and detection. So our approach has been to take that derivatization step and move it into the gas phase. And I'll go into why we think that's beneficial in just a moment. But essentially now we're generating the ions from the tissue surface just like we would in any other imaging experiment, storing them inside the mass spectrometer, doing some type of gas phase transformation or reaction, and then detecting the products. And so the general premise here is that instead of just using the mass spectrometer simply as a detector of molecular mass, we're using it as a reaction vessel to enable unique chemistry. And so ion-ion chemistry has been around for quite some time now. Perhaps the most well-known type of ion-ion reaction is electron transfer dissociation. Obviously, this is widely used in the proteomics community to do peptide sequencing. And proton transfer reactions, which were first discovered a few decades ago, have seen renewed interest in recent years. These are now being used to nicely simplify and kind of decipher out mixtures of intact proteins. But I would argue that these 2 types of reactions, electron transfer and proton transfer, are relatively simple ion-ion reactions. So these are just single particle transfer events. And so if we start to consider other types of reactions that we can perform in the gas phase, things like metal ion transfer or multiple metal ion transfer, or even complex formation or an organic synthesis, we can be more sophisticated in the types of chemistries that we exploit in the gas phase. Okay, so why use ion-ion reactions? I mentioned that I would discuss, especially in related to imaging, why this is so beneficial or why we think this has merit in these types of workflows. And the first reason is that we're eliminating any physical manipulation of the tissue. So we're doing all this derivatization in the gas phase, so we're not adding an extra step to our sample preparation. And aside from kind of saving time, this is really important because anytime we manipulate or treat the tissue, we have the potential to introduce artifacts, right? And so that's obviously a detriment to any workflow. So the less that we can kind of perturb the original tissue sample, the better off we're going to be. Additionally, these are highly exothermic. So when I say ion-ion reaction, I'm referring to reactions between positively charged and negatively charged ions. So there's a tremendous Coulombic recombination energy associated with that reaction. So that means that we can drive these reactions to occur really, really quickly in the gas phase. So these typically occur on the hundreds of millisecond time scale. And if we consider that or contrast that with a condensed phase derivatization, which may take minutes or hours or even days to perform an incubation and reaction, these are much, much faster. We also have really precise control over the reactants and the reactions. So these are occurring inside the mass spectrometer. So everything's under computer control. We have the ability to isolate the reactants using a quadruple mass filter, and we can control the reaction timing really precisely. So we get a really clean product. We can minimize any side reactions pretty well. And this obviously can't be done in the condensed phase in the tissue or in solution. So we tend to end up with a cleaner product. It's also easy to compare the derivatized and underivatized samples. Obviously, if we've derivatized the tissue sample, it's very difficult to then underivatize that sample and recover the original tissue sample. If we're doing this derivatization step in the gas phase, we can simply stop admitting our derivatization agent to the mass spectrometer, and we can just look at our tissue in its native form, underivatized. And additionally, I won't talk about this here today, but we have the opportunity to access new chemistries in the gas phase. So because there isn't a pH in the gas phase, really, we can see some really unique reactivities that we would be unable to see typically in solution. Okay, so this is how we're doing our experiments, our gas phase ion-ion reactions. These are all performed in a Bruker Celerix 7 Tesla hybrid FTICR mass spectrometer. And so our experiments start by injecting our analyte cations, at least for the reactions I'll show to start today. We're injecting analyte cations from our MALDI source through our quadrupole mass filter and storing them in a hexapole ion trap inside the instrument. This process takes a few hundred milliseconds, maybe 200 milliseconds or so. Then we introduce our reagent anions generated via electrospray. We pass those through our quadrupole mass filter and store those in the hexapole ion trap. And this process takes maybe 50 milliseconds or so. And then we let these react for some period of time. Typically, this is going to be anywhere from 500 milliseconds to in excess of a second. And then we can transfer the products to the ICR cell, which is kind of off to the right of the screen here, and perform mass analysis. Since this is an imaging experiment, we obviously want to minimize any downtime to the experiment. And this is a hybrid instrument, so we actually have the ability to kind of nest our reaction. So we can perform the upfront reaction in this region of the mass spectrometer while we're doing the mass analysis in the ICR cell. So in theory, we could basically minimize any detrimental effect to our duty cycle or our experimental throughput. Also, all the experiments that I'm talking about here today are what we call charge inversion ion-ion reactions. So for the first set of experiments, I'll talk about starting with singly charged cation. So this is an analyte that's positively charged, and we're reacting that with a doubly deprotonated reagent anion. And then, so what that process does is it takes our cation and it transforms that into a negative anion. So this is basically that charge inversion process, going from a positive to a negative ion. Okay, so why would this be useful? Here's just a quick explanation of this process, at least as it applies to phosphatidylcholine lipids. So when we're doing MALDI mass spectrometry analysis in positive ion mode for PCs, we readily observe sodiated, potassiated, as well as protonated ion types. And in negative ion mode, we readily generate deprotonated ions. And when we perform tandem mass spectrometry just via conventional CID on these ion types in positive ion mode, we tend to get cleavages around the phosphatidylcholine head group. And so that allows us to confirm this lipid as a phosphatidylcholine. In negative ion mode, we get cleavage at the fatty acid or ketene position. And so that allows us to identify the identities as well as the positions of these SN1 and SN2 fatty acyl tails. When we perform this analysis from tissue, so in a much more complex chemical environment than just synthetic standards, the story is the same in positive ion modes. We generate roughly the same ion types. And when we perform CID on these analytes from tissue, we get similar chemical information. But in negative ion mode, we don't tend to generate deprotonated phosphatidylcholines. And that's because we tend to observe ionization suppression from our much more acidic glycerophospholipids, things like phosphatidylserine, ethanolamine, and inositol, as well as a few others. And so it can be difficult for us to identify the fatty acyl tails of phosphatidylcholine lipids directly from tissue. And so what we're going to use is an ion-ion reaction to facilitate this identification. And the general premise that we're relying on here is that the structural information we get from a tandem mass spectrometry experiment is a function of two main variables. The first is the dissociation method. So are we using electrons or surfaces or photons or collisions to impart energy into the system? And then the second is the ion type. So do we have a protonated ion? Do we have a deprotonated ion? Is it multiply charged? Is it sodiated? Is it a radical cation or anion? And so our experiments here are going to seek to manipulate the ion type in the gas phase prior to doing the dissociation step. Okay, so this is kind of a chemical reaction workflow of this charge inversion process. If we have a phosphatidylcholine in positive ion mode and we perform CID on it, again, we only get cleavage around the phosphatidylcholine head group. So that allows us to confirm that this is a phosphatidylcholine. And based on the mass of the precursor ion, we can identify that it has 34 carbons in the SN1 and SN2 tails and 2 double bonds. So what our solution is to transform this into the negative ion mode. And so to do this, we take our singly charged cation and react it with a doubly charged phenylene dipropionic acid reagent dianion. And this is chemistry that was first worked out while I was in Scott's lab by a colleague of mine, John Stutzman, in collaboration with Steve Blanksby. And so these two ions form a complex with one another. And the reaction proceeds via a proton transfer and a methyl transfer from the PC head group to result or produce a demethylated PC anion. So we've gone from a positively charged ion, and we've gone through this charge inversion process to produce this demethylated ion. And now that we're, excuse me, in negative ion mode, when we perform collision-induced dissociation, we readily generate fragment ions that are indicative of the SN1 and SN2 fatty acyl tails. And so let's go through a couple examples of that. So here's an example of the phosphatidylcholine lipids that we would detect from a typical rat brain imaging experiment. And we see protonated, sodiated, castated ions, a wide variety of PC fatty acyl content. And so let's take one of these. We'll take PC34-1. Obviously, if we do CID in positive ion mode, as I mentioned, we really largely only generate that phosphatidylcholine fragment ion, so it doesn't give us any information about the SN1 and SN2 fatty acyl tails. So our solution again is to take that PC34-1 precursor ion, react it with doubly deprotonated PDPA, and this produces that demethylated product ion. And now when we perform CID on that demethylated product ion, we can readily generate the SN1 and SN2 fatty acyl tails. And I should point out, this is really great work done by Jonathan Specker, a graduate student in my lab. And so upon first inspection, we would think that the 18-1 fatty acid would be present at the SN2 position. And that's because we know cleavage at that position is much more facile. And so we would hypothesize that this would be PC16-0-18-1. But we know that we're making these measurements from tissue and that this is likely a really complex chemical environment. And so what Jonathan did was he wanted to generate a calibration curve where he mixed different proportions of these two synthetic standards, PC18-1-16-0 and PC16-0-18-1, so that we could calibrate this ratio and determine exactly how much of each isomer was present in the tissue. And so when Jonathan did that experiment in different regions of the tissue and applied that calibration curve, we saw what I'm showing here, this bar graph on the left. And I think that this is striking for two main reasons. The first is that both isomers are present in significant abundance. So they're present in at least 25% abundance or so. And additionally, the relative abundance of each of those isomers differs or changes depending on where we are in the rat brain. So whether we're in the granular layer, the cortex, the white matter, the corpus callosum, et cetera. And so I think obviously this has to do with either the biosynthetic origins or metabolism of these lipids, and likely has important implications in their functionality in different regions or cell types in the tissue. Importantly, our work agrees quite nicely with some work that was done a couple of years ago by Steve Blanksby, Shane Ellis, and Ron Heeran, where they used ozone-induced dissociation, so an ion molecule reaction, to perform a very similar type of experiment. This initial OzID experiment required 10-second per pixel reaction times. I know Shane has shown some really nice data where he's been able to perform this OzID experiment in a high pressure environment in a certain region of an instrument. And so that dramatically improved their reaction time and their throughput, because obviously 10 seconds per pixel is going to require a pretty significant analysis time. But we think that the ion-ion reaction inherently should be much faster just because of that Coulombic recombination energy. But in general, I think both of these reactions, ion-ion and ion molecule, are pretty complementary, and there's applications where you would definitely want to do one or the other depending on your experimental needs. Okay, so that was PC34-1. Let's look at another lipid, PC36-1. So again, we've done the ion-ion reaction. We've generated this charge-inverted demethylated product, PC anion. And when we perform CID on this anion, we see that this is now a mixture of 4 different ions. So we have PC18-1-18-0, PC18-0-18-1, PC16-0-21, and PC21-16-0. And we're still working on kind of calibrating the relative abundances in each of these different regions of the rat brain. But again, these lipids are each present in pretty significant abundance, and their variations, their abundances do appear to vary depending where we are in the rat brain tissue. Okay, so one final example, PC36-2. Again, we've done our ion-ion reaction and generated this demethylated product ion. And when we perform CID on this, we see that this is a mixture of at least 5 different isomers. And so we're saying 5 different isomers at least here because we've said nothing about the double bond positions, right? We're only talking about the SN1 and SN2 fatty acid positions. If we consider the positions of the double bonds, are they in the delta 9 or delta 11 or delta 7 position, we could be talking about 5, 10, even 20 different isomers. And these are all isomers that are underlying that single peak, that single PC peak in our MS1 experiment. And so obviously the failure to resolve these is going to hurt our ability to make any meaningful conclusions from some of our imaging mass spectrometry experiments. So we think that this is a nice example of how one way that we could differentiate some of these compounds. Okay, so another nice benefit of this type of reaction is that it also affords us the ability to concentrate ion signal. So it turns out that protonated, sodiated, and potassiated ions all react via similar mechanisms with this PDPA dianion. So they all result in a charge-bearing particle transfer as well as a methyl transfer to the PDPA. And so in tissue, we tend to see each of these ions in pretty significant abundance because there's a lot of sodium and potassium that's present in the tissue because of the roles that these ions play in ion channels and different gates and things like that in our cells. So those ions are present, but they tend to dilute the signal for any single phosphatidylcholine into these 3 ion channels. And so what we can do is basically concentrate the diluted ion signal from each of those 3 ion types into a single, I'm showing a deprotonated ion here. It's actually obviously a demethylated ion, but we can concentrate the signal into a single demethylated anion. And so here's kind of a proof of concept experiment showing that. So here we have our protonated PC36-1. This generates our intact complex where the PDPA is still attached. And then we also observe our demethylated anion. And if we isolate our intact complex and perform SORI CID on that, we generate exclusively our demethylated anion. So the major product or the only real product of this complex is to go to the demethylated peak. We added a bit of excess energy to the system to kind of prove that that was the case. And so you see subsequent secondary cleavage to the fatty acyl cleavages. But the first primary step is exclusively this demethylation event. Okay, so for sodium, it's the same story with the sodium ion type. So we generate the intact complex. CID of the complex exclusively produces our demethylated anion. And the same thing with the potassium ion. So we produce the intact complex, and then that fragments to give exclusively our demethylated anion. And so obviously we wanted to not do this on each ion type individually, but the idea is to do this as a mixture. So we took this mixture of phosphatidylcholine ion types, protonated, sodiated, and potassiated, and performed that reaction. You can see that we generate complexes for each one of these ions, and then they all obviously decompose to form our demethylated anion. So this was a nice kind of proof of concept experiment that we're excited about. We're still working out kind of the best activation to use, the best broadband activation to use to fragment these intact complexes and force them all into this demethylated channel. But I think this kind of nicely demonstrates the ability to not only improve our signal to noise ratio in these reactions by eliminating chemical noise, but also by concentrating analytical signal. Okay, so that, I've kind of showed so far an example of using these reactions to separate isomers and allow for their identification, as well as concentrating ion signal, but we can also separate isobars. And so if we consider this peak here at nominal mass 806, it has a certain distribution in this mouse brain tissue section. But if we acquire this mass spectrum at higher spectral resolution, we see that this is actually a mixture of two different ions, one PC363 plus sodium and PC386 plus a proton. And each of these actually has unique spatial distributions, especially if you look kind of in the cerebellum region here at the top of the brain. And so obviously we want to be able to separate these from one another. And so we wanted to use our ion-ion reactions to do this. Again, since each ion-ion reaction reacts via the same pathway, this particle transfer and a demethylation, the resulting demethylated ion is going to vary pretty significantly between these two. And so when we do the ion-ion reaction, these two product ions now differ by about 22 Daltons, or the difference between the charge-bearing particles. And we're able to easily resolve them on any mass spectrometer, right? Not just a high-resolution platform. So this is an example where we've done this experiment for two of the ions individually. And then when we perform that on the mixture of ions, this was done on standards, this is done on the mixture of ions, we obviously see our intact complex as well as demethylation. So in the complex, we're not able to differentiate the two ions from one another, but when we lose the charge-bearing particle as well as the methyl group, these are easily resolved from one another. So that's kind of the basic concept that we're using here is we're trying to differentiate the reactions, or excuse me, the different ion types based on their chemical reactivity here. And up until now, I've really only shown examples where we've made kind of spatially localized measurements. We've looked at the corpus callosum of the rat brain or the cerebellum or the hippocampus, but we really want to perform this at every single pixel. So we want to do this in a true imaging mass spectrometry experiment. And so that's what Jonathan has done here. We've basically taken the population of ions at 810 nominal mass, and we're going to subject them to the imaging, to our imaging mass spectrometry ion-ion workflow. And so we've acquired this data initially on our FTICR at fairly high resolving power. And so we're able to see three different ions, PC384 plus a proton. We're just able to resolve PC361 plus sodium. And we have another potassiated ion that we think is either a phosphatidylcholine or a sphingomyelin. And each of these has their corresponding spatial distribution in the tissue. So that's obviously, we could do that with high resolving power, but that could take a long time. Or what if we don't have the ability to use a high mass resolving platform? We want to use our ion-ion reaction to allow the differentiation of these compounds. So we've taken the some population of ions at M/Z810 and subjected them to our ion-ion reaction. And we can see we generate our intact complex as well as demethylated forms of two of our ion types. And so if we plot the distribution of this intact complex, obviously this is a mixture of all of the isobaric components of that mixture. And then we can generate images for the demethylated ion. So we're readily able to generate a unique image for PC384, the demethylated form of that. And while I'm showing that we're able to detect demethylated PC361, we actually weren't really able to generate that in significant or in enough abundance to generate a true ion image for that. And so this was, we were excited about this initially, but then, we were worried that we weren't going to have the efficiency in this reaction to do a true imaging experiment. And we've since improved on this. And so we're continuing to kind of work at this. But alternatively, we could also use other components in the spectrum to generate ion images for each individual lipid species. And so what do I mean when I say that? So in general, I showed this charge inversion workflow a minute ago. So we have our protonated or sodiated or potassiated analyte cation, and we produce our demethylated charge inverted PC anion. So that's one reaction pathway that this ion-ion reaction could proceed through. It turns out there's also a competing reaction pathway where we just have a single particle transfer from our phosphatidylcholine to the PDPA. So that results in a neutralized PC that we're not going to detect. and a charge-reduced PDPA anion. And so that's what all of these compounds down here are. And so instead, we can use these particle transfer events, this proton, sodium, and potassium particle transfer, as kind of the other dance partners for this reaction. So we can use these as proxy measurements for each of these individual precursor ions of interest. And so that's what we've done, and we've been able to then plot the spatial distribution of each of these three peaks in our product ion spectrum, and they show nicely the same distributions as the precursor ions. I'll also point out that there's this little stripe here in each of these images, and we actually kind of did that on purpose. So we wanted to convince ourselves that all of these ions that we were observing in our product ion spectrum were actually due to the ion-ion reaction itself and not some artifact of other ions being injected or produced in the system. So during each of these, we stopped admitting the electrospray reagent for a certain period of time and then turned it back on after a set period of time. So obviously we lost or dropped out of the ion signal during that removal of the reagent. Okay, so those are some examples with PDPA. I thought I'd just close with showing one or two other quick examples of reactions that we're exploring in my group. So obviously the PDPA reaction was pretty exclusively restricted to identifying SN chain identity and position. We're also exploring ion-ion reactions that allow us to identify double bond position. So this is also a nice benefit of ozone-induced dissociation that I didn't really discuss, but we want to explore some ion-ion reaction capabilities in this space. And so this is some chemistry that is also based on some work by Scott McCluskey and Steve Blanksby. And what we're doing here is we're taking fatty acid anions and performing a charge inversion experiment going in the opposite direction now, so going from the negative ion mode to the positive ion mode, and then performing collision-induced dissociation. And the chemistry that we're using here is magnesium trisphenanthroline chemistry. So essentially what we're doing is we're adding a ligated magnesium ion to the system. And this magnesium ion, once it's added to our fatty acid anion, when we perform collision-induced dissociation on that, that essentially locks down the charge of the fatty acid carboxylic acid group. And so if you've ever done CID on underivatized fatty acids, you tend to see carbon dioxide, carbon monoxide, just kind of uninformative neutral losses associated with the head group. But if we lock down that charge, we tend to see much more charge remote fragmentation. And this kind of charge remote fragmentation was first observed decades ago by Mike Gross and other people when they were using high energy CID to enable charge remote fragmentation. And so by locking down the charge, that's now what we're able to do. And so we tend to see then carbon-carbon bond cleavage along the fatty acid backbone, except at the double bond positions. And so this work is being done by Julia Bonney, another great graduate student in the lab. And so Julia has taken fatty acids, reacted singly deprotonated fatty acids with doubly protonated magnesium trisphenanthroline complexes. These things generate complexes quite nicely, very efficiently. We don't have any kind of single particle transfer competing reaction pathways, which is nice. And then we're able to perform CID on those these coordinated fatty acid groups. And again, we tend to see kind of a dip in intensity around where the double bond should be. And so Julia has done this for a number of different fatty acids. Here's an example where she's doing this on two different isomers, the delta 9 and delta 11 isomers of 18-1 fatty acids. And you can see in the 18-1 fatty acid, we have cleavage at the H and K positions, so positions kind of surrounding the double bond, roughly in allylic positions. But we don't see any cleavage at the I position, which is right where the double bond is. Conversely, in vicinic acid, we do see that I ion type, but we now don't see the K ion because that is at the double bond position. So we can use both the I and the K ions as kind of markers or ions that basically signify the presence of one isomer or the other. So Julia has also made these measurements from tissue. The efficiency of this reaction, at least on the dissociation side, is something that we're still optimizing. So you can see that the signal to noise on these spectra are quite low. But nonetheless, from tissue, Julia was able to detect both the I and K type fragment ions. So we're not able to quantitate the relative isomers yet in tissue with this approach, but we do know that they're at least both present. And so this is some exciting work that we're still working on. Obviously, you would say, okay, that's a pretty subtle chemical change. Why would we care? Does that really matter? And there's been some really nice data shown by Usha's group that basically says that this can be really important, especially in certain cancers. So Usha is showing here differences in that same fatty acid, 18-1, looking at delta 9 and delta 11 isomers. And she observed that the relative amount of the delta 11 isomer, which again just differs by the single position of a double bond, has a dramatic implication on the relative abundance of that isomer in normal versus cancerous breast cancer tissue. So these are subtle chemical differences, but they're obviously really important to tissue biochemistry and disease pathophysiology. Okay, so I also wanted to close and say, we're looking for ways to more efficiently dissociate some of our complexes. We've been kind of limited to SORI CID at the moment, which isn't the most efficient, and it takes a long time. We're obviously pulsing in gas to our ICR cell, and we need to wait for that gas pulse to subside before we can do the next experiment. So the SORI CID form of the ion-ion reaction is not really amendable to imaging mass spectrometry timescales. So Julia and Jonathan, in collaboration with Wu Young, are implementing an IRMPD laser onto our system so that we can do IRMPD on our product ions. This should give us much more efficient dissociation, as well as allow for these experiments on much more rapid timescales. And this is something they've just done in the past couple weeks or so. And our first kind of spectrum, that we've acquired from this shows that IRMPD is indeed capable of making these dissociating these complexes much more efficiently. And so they've done this for one of our PC-PDPA reactions so far. Okay, so with that, I'll just wrap up and say that, despite the advances in mass spectrometry in general, and specifically in the mass spectrometry field over the past few decades, new technologies are still really critical and crucial to help us address increasingly complicated biological and clinical hypotheses. Hopefully, I've also been able to show you that ion traps can be used as reaction vessels to enable unique chemistries. And a few decades ago, these types of reactions may have been a little bit more esoteric or harder to come by, but ion-ion reactions are increasingly available on a wide variety of commercial platforms. So the commercialization of ETD and some of these proton transfer reactions means that we can implement more sophisticated gas phase reactions with a lot less instrumentation or modification than we could once upon a time. Importantly, these are also ionization agnostic. So I've shown examples where we're using MALDI-generated analytes in our reactions. We could also easily do this with DESI-generated ions, ESI, APCI. It doesn't really matter how we've generated the ions, right? We're doing the reaction in the gas phase. So we generate them however we'd like, and then we can perform some unique and what I think is fun chemistry. Additionally, I've shown that these are applicable in imaging experiments, but any type of online experiment where time is a concern, or we want to transform the ion type after we've generated it, so things like LCM-SMS experiments could also be applicable. And hopefully I've also shown you that gas phase ion-ion chemistries are fast and efficient. They provide a high degree of molecular specificity without requiring a high mass resolution analyzer. And we can also improve our sensitivity by not only concentrating ion signal, but also by removing chemical noise. So with that, I'll just conclude. Thank again the graduate students who performed all this work, Julia, Wu Young, and Jonathan, folks at Bruker for their help in kind of getting these ion-ion reactions to work on our system, specifically Steve Van Orden and Mark Ridgway, the NIH for funding, and you for your attention. I'd be happy to answer any questions that you have.
All right, that was fantastic. Thank you very much, Boone. As the person who's moderating, I get the ability to ask my questions first. So I'll start with two. One of the things, I come from a proteomics background, and the analogous experiment that we think about proteomics is when you think of histones and post-translational modifications, where there are gajillions of possibilities when you do the calculations. But the reality is there's only probably 400 or 500 that actually exist that are doing these sorts of things. Do you envision the same situation is happening with lipids?
I think the answer is absolutely. Yeah, I mean, if we consider, like, if we just consider double bond position, right? We know that things like the delta 9 and delta 11, delta 7 position are relatively common. The even position double bonds are pretty rare, at least in mammalian systems. The odd chain fatty acids are pretty rare, right? So we can just, based on likelihood, eliminate a number of mathematical possibilities. And then I think, again, some of those just tend to be more abundant. So, you're not going to see 100 or 100,000, right, in theory. But as I showed in that one example, you are going to see at least 5 in terms of SM position, and probably in some cases, maybe a dozen if we consider all the possibilities. And I think they are going to be present in relatively significant abundance. So yeah, it's not going to be 100,000, right? But it is it's greater than 1, and a lot greater than 1 in a lot of cases.
And I'll finish with this one and then pass it around. Along the same lines of that, all this new technology is being based on increasing our separation efficiency, especially with things like high resolution, obviously FTICRs, things like slim devices in the beginning, things like TIMS devices. You're working with Bruker a lot. How much is too much in that, right? You think about a TIM cell is only 5 centimeters. If you make it a meter, you get like unbelievable amounts of separation. But when does it become? Are we doing enough for this? And I'm thinking also of those new folded TOF instruments that have come from Waters and all these things that are just made for imaging. I don't know. Is it in the right direction, or is it still not far enough, do you think?
Yeah, I mean, I think the answer is, there's no one universal tool, right? There's not going to be a single approach, a single instrumental platform that's going to work in all cases. So I think the more tools that we have available to us, the better we're able to address any one challenge that we may face. So I think that would be the general answer I would give, but I don't think we're there yet. We're still, when I show some of this data to some of our biological and clinical collaborators, frankly, the first time I did, they didn't know what to do with it. They, when we, as mass spectrometrists, show them that when you look at phosphatidylcholines, there's already, a few hundred things that we detect. And then we tell them that there's several isomers for each one of these things. Biochemically, that's just not a dimension that a lot of folks, I think, have thought about. So I think it's still finding utility. I think it's, again, going to depend on the application. If you're talking about something that's really going to be in a clinical setting, right, then the translational bridge is not going to be able to carry a $1.5 million FTICR over it. It's not that strong, right? Speed is much more of a concern in that space. Identification maybe isn't as much of a concern, maybe just the ability to separate something. So maybe ion mobility finds a better utility there. So I think the strengths and weaknesses of each of these things can complement each other. You can use them in tandem and use them in different situations.
Very cool. I'll pass it on to everyone else.
Yeah, I got a bunch of questions. I think that's a good answer. So not the tool, but also the individuals who are running this, right? I mean, the complexity of the device itself. I mean, we need different tools for the purpose of, how the usability in mind, right, as well. So maybe a combination of all these different technologies are useful, just not for the purpose of developing new technologies, but also keeping the user in mind, right? These ion chemistries are pretty novel, and its widespread usability is somewhat still limited, right? Mostly in academic settings, except for the well-known reaction, which is electron transfer, and now the proton transfer, PTCR capabilities on the Eclipse, right? So going back to your PDPA group transfer, right? Methyl transfer. So this is a dianion transfer of methyl groups. Is there any limit to the size of the group transfer? Have anybody tried, larger group transfers than methyl?
Yeah, that's a great question. So for lipids, no, we've just kind of done, a lot of the lipids that we're going to look at in positive bimotor are going to be sphingomyelins and PC. So they just have, 3 little methyl groups off that quaternary ammonium. But we have looked at this when I was a grad student in Scott's lab. This was a reaction that Josh Gilbert and I started looking at, and we looked at methyl transfers, ethyl, butyl, propyl. You know, we just looked at the ability to transfer a ton of these different alkyl chains to whatever dianion that we were using, and we used a lot of diacids. We looked at EDTA, right, and all these different types of things, and we were able to transfer quite large alkyl groups, and so it did become more sophisticated or elaborate, the types of things that we were able to transfer. So it definitely is possible.
Yeah, I mean, going back to the fundamentals, I mean, there is some limitation as to the size of the reaction, right? I mean, if you look at the kinetics and the amount of energy that's deposited in that kind of reaction, the larger the system, you know, the amount of energy that really gets localized on those bonds for rearrangement is somewhat limited. Has there been any kind of fundamental understanding on, you know, what those limits are? And if there are, is there some way that we can increase the enthalpy of the reaction in some ways to facilitate those reactions? I mean, since you come from that background, I thought, you know, that would be something of somewhat useful thing to think through for, you know, future reactions, right? If somebody was to do something completely different, not look at lipids, but look at other types of molecules that require these types of transformations, can one predict those and actually build instruments to perform those experiments? Yeah,
basically more intelligent reagent ion selection, right, based on the theory that you would predict. The short answer is yes, it's possible. We did a little bit of this when I was leaving Scotch Group. We published a paper where we looked at reaction exothermicities as predicted by things like gas phase basicities and things like that, and it did track well. So it was something that we were able to do, and it's absolutely something that we're going to start to move into now. We picked PDPA because as a first-year professor, I wanted something that I knew that would work, and so that's where we started. But already we've seen differences between the reaction performed on our system and the reaction that John performed in Scott's lab, and that was done on a Q-Trap 4000. And so there's, the reaction exothermicity should be the same on those systems, right? So the differences then the differences explicitly that we saw were the relative ratio of intact complex formation to spontaneous demethylation. And we think that that is possibly due to a number of things. It could be due to the energetic transfer conditions between our hexapole and our ICR cell of the product ions. It also could be a pressure dependence. So the pressure in our hexapole is a lot different. The pressure is higher, it's going to dissipate energy more quickly. And so I think that that probably is going to play a big role. So we absolutely think that there's ways that we can more intelligently not only pick our reagents, but set up the reaction conditions for each individual reaction.
Okay. One more quick question, if there are no other questions. Why don't we go around first before I ask questions? Silence. All right, let me ask them. So you talked about competing channels, right? So what is the efficiency of your charge inversion reaction, the metal transfer reaction?
Yeah, it's a great question. And we haven't quantified it exactly for a number of reasons. Obviously, positive and negative ion efficiency, those detections are going to be a little bit different. So we need to kind of take that into account before we do that accurately. But we can say that the relative efficiency for the proton, sodium, and potassium is not the same. So we know that the single proton transfer or the proton and methyl transfer is much more efficient than trying to transfer the larger cations. So those reactions seem to favor the single particle transfer without the methyl, so just the charge reduced PDPA. So we know that there's different efficiencies depending on the charge bearing particle. And that just kind of makes sense, right? It's going to be harder for one of those big things to hop over. So we're still working on the total efficiency. We're still working on exploring the kinetics, because we think that we can probably not only differentiate the products based on the mass, but also based on the kinetics of the reaction. So if we look at the relative kinetic rate of formation of, the sodiated versus the potassiated ion, for example, we should be able to identify or differentiate isomers in that respect too.
So the PDPA is a dianion. when I was in the lab, we used different types of dendrimers to do multiple proton transfers for charge inversion.
Yep.
Is there any reason why you are not doing that, or is there some reasoning behind choosing the right type of anion to do this inversion?
Yeah, so we're exploring dendrimers too. It's a great question. And that was right around your time when all that was doing. We've actually tried to do a little bit of Kelly Newton's work, where I think she was using dendrimers to transfer some metal ions. So that's kind of an alternative that we were exploring to this magnesium trisphonanthrene chemistry. So we're absolutely looking at some of those. The reason that we've selected PDPA, at least to begin with, is on our system, we're seeing a little bit more sensitivity to the disparate masses of the reactants than we saw on some of the systems that we worked on in the McCluskey lab. So it's a little bit harder for us to mutually trap things that can be quite different in M over Z. So, if we're looking at a fatty acid or a lipid and we have a little EDTA or PDPA, it tends to be a lot easier for us once we start getting higher in products, especially when we have a charge reduced product. That is being a little bit more challenging for us. So that's why we stuck with PDPA to begin with, but definitely something that we're going to explore.
One final question about usability, right? I mean, these are pretty sophisticated instrumentation that you're talking about, right? If we were to have this commercialized, if you're thinking lines of, how we get this to this instrumentation to the masses, how they would be using this, you had a single source for introducing both reagents and the lipids, right? Is there an advantage of having multiple sources or entry points to do these, so that you disengage, you do things, maybe the imaging experiment has some benefits, some merits to doing that way, or do you see any kind of advantage to designing instrumentation with multiple ion sources, or just having just single entry?
Yeah, I mean, I think the multiple sources provide us with a lot of flexibility from a research perspective, right? But if we're talking about streamlining it for something that would be more adoptable to the masses, definitely, I think, Harsha, you did a lot of the preliminary ETD work, right, in the group. And so, if I were to ask you that question,
long time ago.
yeah, if I were to ask you some of those questions about ETD, like, back then it was a much more sophisticated experiment, much less integrated on systems. So I think as that hardware, the ETD hardware, is becoming more ingrained and commercialized, I think we can exploit some of those things. I think nowadays most of those ETD sources are CI sources, right? They're not dual sources at the front of the instrument anymore. And so if we can, that's something we'd like to do. We have an ETD source actually in our instrument that we don't use. So we actually have 3 sources. So we'd like to explore the ability to use the CI source to generate some of these reagents. We'd also like to explore, additional ion molecule reactions. So that's not something that we've done a ton of to this point. It's something some others have done, but those types of modifications are much easier, right? We're just introducing a neutral reagent into the collision cell or something like that. So I think, yeah, there's some benefit to a more simplified setup, but I think the hardware will be there, right? Same thing with UVPD, right? If Joe, if we're talking about Jenny's lab, right? 20 years ago, that was some crazy stuff that she was doing, right? But now UVPD, you can buy a thermo instrument, you can buy a couple instruments now that have UVPD. So I think it's just becoming more available.
And if I that's a perfect segue to what I think will probably be the last question, unless anyone else has something afterward. One thing, I noticed that really cool magnesium reaction where you chelate the magnesium between those two I forget what they're called, but you put them together and those look remarkably like photocatalysts. And so this is one of the things that makes me wonder, if you put a BPY ligand in there, and I've been thinking about this a lot since the Nobel Prize was just awarded to a photochemist. If you put that in there and then shine the right light in there, you could start getting some really cool, like dexter energy transfer type reactions and things like that. Because I know there have been a couple publications that have talked about observing gas phase fret in an ICR cell. So it could be really interesting to just add, like Harsha was saying, something totally new and different with the capabilities that you have there.
Yeah, and that's something that we're doing. We have a UV laser that we're going to be implementing in addition to the IR laser back there. And you're exactly right. I mean, there's some chemistry that we first worked out when I was in Scott's lab, where we could use sulfone HS reagents to add AMCA or different coumarin rings or things like that, right? And you can just, or it could be something like the phenanthroline, right? And these super UV active things could enable some unique chemistry. So that's that other half of those two beakers, right? You know, you have the ion type and the dissociation technique. So we're definitely looking at mixtures of all those different types of things. Some of the things we could probably solve with just UVPD, right? You know, Jenny's doing some really nice work where she can just use UVPD and identify double bond positions. But I think that there's some potential there where we can still use both, you know, both an ion-ion reaction and some type of photo dissociation. We're also doing electron-based dissociations like EID, EIEIO, like Ben Freischer called it, you know, 50 years ago or whatever it was. So I think there's a lot of phase-based.
Now it's called electron activated dissociation. They rechristened that term.
Yeah.
So
Exactly.
SAG took over that term, and they're calling it EAD.
Yeah.
They're calling it the new form of dissociation.
Yep.
New ECD.
Yeah, that's right.
About 30 years old.
Yeah. Yeah. So I think there's just a lot of phase space, right, in this kind of gas-phase chemistry world that we get to play in, which is a lot of fun.
That's fantastic. I see we're at 6:15. If anyone has one last burning question, maybe we could take it. Going once, twice, on. All right. On behalf of everyone here, I want to say thank you, Boone. That was an excellent talk. We really enjoyed it, and we're going to keep an eye out for more great work coming out of the Prentice Lab.
Thank you.
Excellent talk.
Thanks, everyone. Bye-bye.
Bye.